So hello everybody, I'm Fabien Maury. I'm a PhD student from Paris, and today I'm going to talk about part of my PhD project, which is to integrate knowledge related to autoantibodies and autoimmune disorders in an ontology that we will populate using data from the literature. So to quickly introduce the topic of autoimmunity, well, in healthy people, part of the way our immune system defends us against pathogens is to produce antibodies. But in some cases, the immune system will produce antibodies against molecules from the self. And in that case, we talk about autoantibodies. And this is what happens in the case of autoimmune pathologies, such as diabetes type 1 or lupus, which you might have heard of if you've watched the House series, where it's some kind of a recurring joke. So there are many autoantibodies. Over 200 of them are currently known. But to our knowledge, there is no single exhaustive list of them. They are involved in many autoimmune diseases, and autoimmune diseases are a broad, diverse group of pathologies which may present very diverse symptoms, which may also be very vague, very general. So it's quite complicated sometimes for medical doctors to diagnose these disorders. Even more so since many of these disorders are rare diseases. So that means there is not a lot of data about them, not a lot of patients. And for all these reasons, sometimes it might be hard for healthcare professionals to input their patients' phenotypes into medical IT systems. Sometimes there is just not the right case to tick to describe the phenotype. So the medical doctors cannot be as precise as they would like to be. And there is a loss of information for this reason. So there is no centralized single source of knowledge which would gather everything we know about autoantibodies. So that's the situation we would like to improve. So our goals are first to gather scattered data online and from different experts teams in a single centralized resource. But we want also to structure this data so that useful knowledge can be extracted from the data and be used in clinical care and by researchers. Then we also want to standardize the way people refer to all these autoimmunity-related concepts, because sometimes some of them are very rare, very new, very niche. So that means one way to refer to an autoantibody could be known only in a single team sometimes, or might coexist with a different name. And sometimes the differences between these different names are not very clear. So we want to standardize all these things. We want to make all this knowledge accessible to the broader scientific community and not only to the experts of these pathologies. So this is our project, the Macao project. And we want to create a resource about the relations between autoantibodies, the autoimmune disorders they're related to, the targets of these autoantibodies, because sometimes they're not always precisely known. And so the way we started this project is we contacted the rare disease networks in France, and we asked them to share all the autoantibodies they knew about. So we got a list of names. Then we also looked into the HPO, the Human Phenotype Ontology, because it has some phenotypes related to the positivity to a specific autoantibody. We parsed this and we got other names. So we had a big list of autoantibodies names, and we gave this list to curators, mostly pharmacists and biologists, and we asked them to go online and look for everything they could find about these autoantibodies, and also new ones if they would stumble upon them. So this is what they did. They got information about more than 350 autoantibodies. They got information about their name, their synonyms when there are synonyms, both in English and French. they got information about the categories of these autoantibodies, because sometimes they group together, they're related. So we have a hierarchy. We also collected the target molecules as precisely as possible for each autoantibody. And we collected the diseases related to them. And as much as possible, we asked the curators to collect the provenance for this information. So that means they had to write down a link to some online resource, preferably from the literatures, but not only. It could be some books, some commercial websites selling antibodies, anything. But they had to write it down. And from these data sets, we tried to put it in an ontology to describe all the relations between the autoimmune diseases themselves, the autoantibodies, the targets of the autoantibodies, and also the clinical signs and phenotypes they might be related to. Because an ontology is a good way to represent some concepts from a field and the relations between these concepts as precise as they might be. So this is what we did. We designed an ontology based on the FAIR semantic web principles. So it can be reused and integrated in other systems. And we populated this ontology using our data. So we also reused in the ontology as much as possible identifiers from other popular biomedical terminologies and ontologies, such as the HPO, orphanet for rare diseases, UniProt for protein, etc. So here is the whole diagram of our ontology. It might be a bit complicated to read, so I will zoom in on a few specific parts to describe them. So we reused mostly orphanet identifiers for diseases because we are dealing with a lot of rare diseases. But we also reuse SNOMED city identifiers or UMLS identifiers. Then we made all these diseases also of the type autoimmune disease that we create. For targets of autoantibodies, we mostly use UniProt identifiers because most of the targets are proteins, but not always. And in the case we have targets which are not proteins, we can use KEBI identifiers or also UMLS identifiers. And we make all these items of the type target that we define. For autoantibodies, we create a class autoantibody. And for each autoantibody we have in our knowledge graph, we link it to a positivity phenotype because the HPO contains items that were antibody positivity phenotypes, not antibodies. And we thought it would be nice to link to the HPO. So we automatically link each autoantibody in our graph to a phenotype of positivity by reusing HPO identifiers when they exist. But if we are dealing with an autoantibody that do not have an identifier corresponding in the HPO, then we create a new one. And we also use HPO terms for symptoms and clinical signs of diseases. So orphanet offers a file mapping diseases to HPO terms, and we parsed that file to collect the HPO terms with the highest occurrence rate for each disease. So we took only the ones with an occurrence rate above 80%. Then I said before that we asked our curators to gather the provenance information for everything they recorded in our dataset. So we also integrate this in the knowledge graph using concepts and predicates from the Provo ontology, which is made to represent this kind of things. So for each relation we want to justify, we create a reified copy of it, and we link it to a document. So a link. The document is can be any kind of online resource, can be a PubMed paper, can be a website or anything else. And we linked these things using the was derived from predicate from Provo. So we have each relation which is derived from a document. Then some people working with autoantibodies talk to us about how it would be nice to incorporate loin codes, especially clinicians. So loin codes are codes to refer to clinical biomedical tests. They are made of several parts. One of these parts is called the component, and it refers to what is being tested for, what the test is trying to detect. So we can have different long codes with the same component. So different tests trying to detect the same thing. And some of these components correspond to autoantibodies. So we tried to match the autoantibodies in our knowledge graph to long components. And we did this using some simple string similarity with their names. And we included these matches in our knowledge graph using SCOS close match predicates. So they're in the knowledge graph, but the matches are also available in a separate file. So this was the whole ontology, but we also implemented a simplified version of it, which relaxes some of the formalism of the semantic web. So here we do not bother with classes and instances of classes. We just link all the entities together using the relevant predicates. We also do not include the provenance information in this simplified version, and we do not include the matches. So it's much lighter. And why did we do this? Well, we thought it would be maybe easier for humans to read, especially people without a computer science background, such as maybe clinicians who would be interested into finding out something about a specific autoantibody. And we made all these things available on a website we are developing. So currently, you can check our datasets on the website. There is a search bar. You can also download the whole file as CSV. You can also download the whole knowledge graph as RGF file. But there is also a Sparkle endpoint if you want to automatically query some things from our knowledge graph, or if you want to integrate some software with our knowledge graph, you can do it. But the website is still in development, and in the future, we will, of course, continue to curate this knowledge graph because we keep having it reviewed by biologists and immunologists. We will also enrich it, and we will try to add some kind of interactive visualization. So still in the same idea to make it easier for people without a computer science background to use it and find information in it. And so you can help for this part. There is a contact form. So if you find something which is which you think is not correct, or if you want to add something, you can tell us and we will try to make the change. So what are the next steps of this project? So we have a manually curated knowledge graph, which is still small. And in the future, we want to try to enrich it using some relation automatically extracted from a large number of texts using natural language processing. So we want to do this from two types of text. First, we collected a large number of PubMed abstracts using the PubMed API to select texts about autoantibodies. And we manually annotated a small subset of these texts for the specific entities and relation that interest us. So these are autoantibodies themselves, their targets, the autoimmune disorders they're related to, and the clinical signs that might be also related to these things. And we also annotated the relations between these concepts. And so we want to use this small annotated subset to fine-tune language models so that they will perform better at the task of detecting these entities and relation in the rest of the texts. So we don't have to do all this manually. This will be done in an automated fashion. Then the second type of text we will be working on are electronic health records. We gained access to a large number of electronic health records from Paris hospital patients, which were selected because they were diagnosed with autoimmune disorders or suspected to be affected with these disorders. So these are both structured and unstructured documents. and we will try to also extract some interesting correlations or relations from this text in an automatic fashion. But this is a bit more challenging because these are documents in the French language, so we will probably not be able to use the same language models as for abstracts. Also, since these are documents from real patients, there is a lot of regulation about privacy, of course. So we only have access to these documents on a specific secure server from the Paris hospitals. We are not able to download these things, and we are not able to work on them using any kind of library. So this is a bit more challenging. So these are the next steps for the very near future. And to sum up, so this is a project to better structure knowledge about autoantibodies and autoimmune disorders. We manually collected a dataset about more than 350 autoantibodies from the literature. We designed an ontology that we populated using data from this dataset. Everything is available on our website, which you can check. And in the future, we want to enrich this knowledge graph using relations automatically extracted from texts of the scientific literature or from electronic health records. So that's it. Thank you for listening. Thank you to my supervisor, Adrien Coulet and Maud de Dieu-le-Veut. Thanks to all the people who contributed to this project, especially for the curation part, which is a very tedious task. And thank you to the people, any people who are collaborating with us on this project, and to the Rare Disease Networks, Brain Team and Filmus, who are funding this project. So you can have a look at the website if you want. And if you have any questions, I will be taking them. Thank you.
So thank you. We have a few questions. Time for a few questions.
Thanks. This is really terrific. As someone who works on autoimmune diseases, this is going to be a really valuable resource. So thanks for putting it together. One question I have is that when you talk about autoantibodies, it's actually polyclonal collection of antibodies that are found in the serum that are detected by serological tests. It's not like a single antibody. So I'm wondering, how are you representing that aspect of that interaction?
So your question is, how do we do in the case the tests are for a whole class of autoantibodies?
Well, it's a collection of antibodies that gives rise to the reactivity. It's not a single antibody. So I'm just wondering how you represent that.
Right now, we would just represent every type of autoantibody. So if there are several of them involved, there would be several relations in the graph.
All right. One more question here.
I appreciate the talk. Thank you for sharing. A very impactful project. I also, just like Richard mentioned, I also work on collecting this kind of data, mainly antibody, and there's a lot of problem and challenges facing. I have a clarifying question. The data that you mentioned, did you only, as of right now, did you only include the data that you received from the Rare Disease Network, or you also curated some data from literature?
So right now, the data in our knowledge graph, what you were asking, the names of autoantibodies from the Rare Disease Networks, this was the starting point, just what we gave to our curators. And then they went online, basically, and they looked for everything they could. So they found things about these autoantibodies that were already named, but they also found things that we did not tell them about at first. They found things that were not in our initial list. And if they collected information about this, then it's in the data set and also in the knowledge graph. But so far, we have not processed PubMed abstracts in an automatic way. This is still being done.
Understood. Thank you.
Okay, so any more question? One last one. A quick one, please.
Thank you. Thanks for the talk. I have a question about your next steps, more technically. You mentioned that you would like to use the manual annotations and then fine-tune a large language model. Have you also considered the traditional methods like BERT-like approach?
The traditional what, sorry?
The traditional ML techniques, the NLP techniques, BERT-like, CyBERT-like models.
Yes. Well, what we are thinking about is also using BERT-based model. So it depends on the people. Sometimes BERT is considered large by some people, sometimes not. But yes, we would also look at BERT-based approaches.
Okay, so please let's thank Fabien again.